I'm Stacey Muse. I'm here with the RRS Podcast interviewing Jai. So would you like to introduce yourself?
Sure. So I'm Jai Smith. I'm a PhD student from Melbourne at the Peter MacCallum Cancer Center. I'm down there under Roger Martin in the radiation biology lab.
Very cool, very cool. So what kind of research do you do there?
So we're in Radioprotection, so pharmaceutical protection of normal tissue is our main area. So we're preclinical drug development at the moment. So our main goal is to develop a small molecule chemical radioprotector for use primarily, ideally in head and neck radiotherapy, for example, normal tissue protection during cancer radiotherapy.
Cool. So why head and neck in particular?
Okay, so head and neck, it's an area where radiotherapy is used quite extensively, but there is some really sensitive, radiosensitive tissues, so mainly the oral mucositis is very susceptible to radiation damage, and that's one of the complicating factors of that therapy. So at the moment, oral mucositis will often limit the amount of radiation that they can give, or it will be responsible for treatment interruptions. So, and as I'm sure you know, with radiotherapy, any interruptions to the treatment can have a really detrimental effect on the success of the treatment. If we can do anything to minimise those side effects, we can improve the treatment. And also, it's just some very big quality of life issues. The kind of ulceration that comes from head and neck radiotherapy can be really painful and uncomfortable for the patients. So that's an important thing to.
Absolutely. Yeah, that sounds like really important work. So can you tell me a bit more specifically about your project with it?
Yeah, so that's the broad project. So we've been in a long-running drug development, so we've got a lot of our own compounds. And what we found a few years ago in a previous project of mine was that when we combine our DNA binding antioxidants, they're called, our particular brand of radioprotector, with the aminothiol radical scavengers, so the amifostine, the classical radioprotectors that have been around for decades now. We actually get additive radioprotection when we put those two together, so which is quite an amazing outcome. We're seeing really high levels of radioprotection. At first we saw that in tissue culture, so that was before my project. And then my project was to see if that phenomenon of the combination radioprotection translates into a in vivo animal model. Okay. And then also I've taken that a bit further, and we're now looking at whether or not we protect from radiation mutagenesis, which is another really important endpoint for radioprotection. Anytime you can increase the survival of cells, you want to make sure that they're still healthy, viable cells, and that you're not increasing the mutagenic changes.
All right, so that sounds like some pretty positive outcomes so far. What are the next steps?
So for my project, I'm actually at the very end, so I'm writing up. So
congratulations.
That'll be closing up for my PhD. But there's definitely the next step for the compound is to advance towards clinical trials, which is something we're in the process of doing. But also the mutagenesis work is really promising. We see a really big reduction, both for the drugs on their own and for the combination. So I think it'll be interesting to take that a bit further into some other models and to test some of our other analogues and just ensure that that's a really robust result. So far I've done a lot of work with one compound, but because this is a fairly untested, it's a very novel compound, and we've got a series. It would be nice to be able to generalise to the entire, or to the broader category of radioprotector instead of to my specific one. So it'd be good to reproduce some of this with some alternative compounds.
Yeah, that's cool. Awesome. So this seems like a really interesting area of research, but how did you actually get into it?
That's a very good question. I think a lot of people in radiation, there's so many different routes to come towards it. Unfortunately, Australia doesn't have an undergrad radiation biology course at the moment that I'm aware of. Definitely not in the unis I was going. So I came in through biochemistry, chemistry as an undergrad. I did that at Swinburne University, and we had a really good program there where undergrads can go and work in a lab for a year, which is, the IBL industry-based learning program, which was a really positive program. So that put me in Professor Martin's laboratory for a year, and then I just got a taste for the radiation science. I think from that I found it really, really fascinating, and it was a really nice way of combining a few different areas of science and using a little bit of my chemistry knowledge and learning some biology and getting hands on some animal models, and it's just such a broad field. You never get bored with radiation, I find. Yeah.
That's very cool. Can you tell me a bit more about that IBL program? It sounds like it'd be really good for a lot of different people.
Yeah, look, it's a great program. So it's basically a paid research assistant role after 2 years of your undergrad. Wow. So before your third year. So you're still really junior when you go into it, and a lot of it depends on who your supervisor is. I think that the unis try and encourage really inclusive, supportive supervisors that are going to kind of foster young scientists and just give people a taste for laboratory life. And it was great. So I did that for a full year, and it's not paid very well, but it's paid. So
enough that you don't have to have another job?
Yeah, exactly.
Okay.
So you can
That's very cool.
focus on your work for a full year, and then you go back to university for your last year, having already worked for a year, which is an amazing help. When you go back to the idea of doing a 6-hour lab in second year is a little bit daunting, and then after working in the lab 5 days a week, the idea of a 6-hour lab's kind of, it's not enough time to get things done. You're kind of like, I could do more. Yeah.
That's cool.
So that was a great program. And then it seems like a lot of people will keep contacts and keep working part-time throughout the rest of their undergrad.
That sounds very cool.
So it's not super common in Australia. It's that one university's really embraced it, and there's a couple of others that have smaller 3-month placement kind of programs. But
hopefully there's similar programs around the world as well.
Hopefully, look, I'd endorse it. I found it to be really helpful.
I mean, it seems to be working out very well for you.
Yes, it really has, and I think it's a great way for labs that don't have that direct undergrad connection, or because we're at a hospital, so we're affiliated with Melbourne Uni, but the Peter MacCallum Cancer Center is separate to the uni, and there's no teaching roles within the university, from the hospital to the university. So it's a really good way for PIs to have access to students that sometimes they wouldn't if they're not teaching directly.
Excellent point.
That's just another route to get people into science and involved.
Very cool. So you said you're just wrapping up your PhD now, and I know you're not really supposed to ask, but any idea what your next steps might be?
That also is a great question. So I'm actively looking for postdoc opportunities at the moment. I'd like to stay in the broad radiation research field, but I'm fairly open as to where. I think radioprotectors has been a fascinating field, and I'd be definitely open to advancing that in some other aspect. But then, I've had a lot of fun going from kind of the more hands-on animal models into a bit more of the molecular biology stuff with mutagenesis. So it's kind of, it's nice to keep your options open and keep exploring.
So you'd probably be sticking around like academia research. Would you ever consider getting into industry or government or any of those other options that PhDs seem to have?
Yeah, look, I wouldn't discount any of those immediately. I think I really enjoy the academic environment. I think that's something really fantastic about that. But we've been lucky enough to have a drug company sponsoring a licensing deal. Mm-hmm. So that's also been a great opportunity to, attend some meetings with, that combination between academic and industry. And I think that's something that's really important. I think it's something that Australia's it's trying to embrace that, getting the industry and the academic world together. But I think there's still a lot of work we can do there to really, take advantage of that. So yeah, look, potentially some role in the overlap between industry and research could be really fascinating too.
So, any advice that you think you could pass on to others based on your experience so far, like what you've liked or disliked about, for example, working with industry or just doing a PhD in general? So, I mean, for people like myself, for example, considering doing PhDs, anything you'd like to?
Yeah, look, oh gee, where to start? There's so much.
Keep it short.
No, I think if you can find a project that you enjoy and find a supervisor that you get along with and that's supportive and knowledgeable in your area, I'd recommend a PhD to anyone. I think it's fantastic, and I think the skills you learn throughout it are great. I think often some of the skills you learn at the end you wish you'd learned at the beginning. So that's I guess that's the advice, where kind of being more proactive and actually thinking how it's all going to come together at the end. It's so easy, especially when you're early in your PhD, just to want to do experiment after experiment after experiment.
Just go right into it.
And generate a heap of data. But then it's not until you kind of take a breath and go, all right, let's go back and look at this data that you kind of realise, I probably could have been more efficient. I could have, organised that in such a way that, you realise you had the answer after 4 experiments, but you did 6 because you were, it was important to generate that data. And so just, I guess, I think if I was to do it over, I'd probably have maintained my enthusiasm, but also kind of take a little bit more of a just keep the considered approach, and easier said than done.
Absolutely. There's always things we'd like to change.
Something that really comes with experience, and
I mean, really, that's what PhD is about, right?
It's about, exactly. It's
Very
learning
cool.
how to be a researcher. Yeah.
Awesome. Any last thoughts or anything before we wrap up?
No, I think that's great. I think we've just moved into a brand new building down in Peter Mac in
nice.
Melbourne. It's an amazing city. I'll put a little plug into Melbourne. We're very proud of our new cancer center, new hospital research, and we've got some really good collaborations with different groups. And we're a long way away from most of the people at this conference from the States and Europe, but we're definitely always looking for collaborations and chances to exchange ideas. So we should come on down to Australia.
Come check it out. That's awesome. Great. Thank you so much.
Well, thank you,
All right. Thanks for coming in.
All right.